Salicylic acid application mitigates oxidative damage and improves the growth performance of barley under drought stress. Introduction. Drought is a severe environmental stress that affects crop productivity and quality worldwide. Barley, Hordeum vulgare, is one of the most important cereal crops and a staple food for millions of people. However, barley is sensitive to drought and suffers from reduced growth, yield and quality under water deficit conditions. Salicylic acid, SA, is a plant hormone that regulates various aspects of plant growth, development and defense. SA has been shown to enhance plant tolerance to various abiotic stresses, such as drought, salinity, heat and cold. SA can modulate plant water status, osmotic adjustment, antioxidant defense, gene expression and signaling pathways under stress conditions. The aim of this article is to explain how SA application mitigates oxidative damage and improves the growth performance of barley under drought stress in an easy and descriptive way. The article is based on the research paper by Islam et al. 2023, which investigated the effects of SA on two barley genotypes, Barley 5 and Barley 7, under drought stress. Materials and methods. The experiment was conducted in a greenhouse at the Institute of Biotechnology and Genetic Engineering, Bangabandhu Sheikh Mujibur Rahman Agricultural University, Bangladesh. The seeds of Barley 5 and Barley 7 were sown in plastic pots filled with soil. The plants were watered regularly, until they were 10 days old. Then, the plants were divided into two groups, control and drought stressed. The control plants were watered daily to maintain 75% soil moisture content, while the drought-stressed plants were watered only once to maintain 7.5% soil moisture content. The drought-stressed plants were further divided into four subgroups, untreated and treated with 0.5, 1.0 or 1.5 m SA. The SA solution was sprayed on the leaves of the plants once every 2 days until the end of the experiment. The untreated plants were sprayed with distilled water as a control. The experiment was arranged in a randomized complete block design with 3 replications. The data were collected after 15 days of drought stress treatment. The following parameters were measured: plant height, leaf area, shoot and root dry weight, relative water content, RWC, proline content, hydrogen peroxide, H2O2, content, superoxide, O2- content, malondialdehyde, MDA, content and antioxidant enzyme activities. Results. The results showed that drought stress caused significant reductions in plant height, leaf area, shoot and root dry weight of both barley genotypes compared to the control plants. However, SA application alleviated the negative effects of drought stress on these parameters by increasing them in both genotypes. The highest values of these parameters were observed in the plants treated with 0.5 m SA. Drought stress also decreased RWC and increased proline content in both barley genotypes compared to the control plants. These changes indicate that the plants tried to adjust their osmotic potential to cope with water loss under drought stress. SA application improved RWC and reduced proline content in both genotypes under drought stress, suggesting that SA enhanced the water status and reduced the osmotic stress of the plants. Drought stress induced oxidative damage in both barley genotypes by increasing H2O2 content, O2- content and MDA content in their leaves compared to the control plants. These compounds are indicators of reactive oxygen species, ROS, accumulation and lipid peroxidation, which can damage cellular structures and functions. SA application reduced H2O2 content, O2- content and MDA content in both genotypes under drought stress, indicating that SA prevented oxidative damage by scavenging ROS. Drought stress also affected the activities of antioxidant enzymes in both barley genotypes compared to the control plants. Antioxidant enzymes are responsible for detoxifying ROS and maintaining cellular redox balance. The activities of superoxide dismutase, SOD, peroxidase, POD, catalase, CAT, ascorbate peroxidase, APX, and glutathione peroxidase, GPX, were measured in this study. SA application increased the activities of these enzymes in both genotypes under drought stress, suggesting that SA enhanced the antioxidant defense system of the plants. Based on the research findings, the following reasonable solutions can be implemented to mitigate the negative effects of drought stress on barley and improve its growth performance. Application of salicylic acid, SA. The research suggests that spraying the leaves of barley plants with SA can be effective in alleviating the negative impacts of drought stress. SA is known to regulate plant water status, osmotic adjustment, and antioxidant defense mechanisms. Therefore, incorporating SA application as a regular practice during drought stress periods can help improve the growth and yield of barley crops. Optimal SA concentration. The study found that treating the barley plants with 0.5 m SA resulted in the highest improvement in various growth parameters. It is essential to determine the optimal concentration of SA for effective results. Further research and experimentation can help refine the appropriate SA concentration specific to different barley genotypes and environmental conditions. Water management. Proper water management techniques should be employed to minimize the impact of drought stress. This can include strategies such as efficient irrigation systems, mulching, and conservation practices. Maintaining soil moisture at an optimal level can help mitigate the negative effects of drought stress and maximize crop productivity. Genetic selection. The research mentioned the use of two barley genotypes, Barley 5 and Barley 7. It is crucial to continue breeding programs and select barley varieties that exhibit improved drought tolerance. By selecting and cultivating drought-tolerant barley genotypes, farmers can reduce the vulnerability of crops to water scarcity and enhance overall yield and quality. Crop rotation and diversification. Implementing crop rotation and diversification practices can contribute to better soil health and water utilization. Alternating barley cultivation with other crops that have different water requirements can help break the cycle of continuous drought stress on the soil and reduce the overall impact on barley crops. Integrated farming systems. Integrating different agricultural practices, such as agroforestry, intercropping, or cover cropping, can provide additional benefits during drought stress. These systems help improve soil structure, increase water holding capacity, and provide partial shade, reducing water evaporation from the soil surface and minimizing drought stress on barley crops. Farm-level water harvesting. Harvesting and storing rainwater or utilizing alternative water sources can provide supplemental irrigation during drought periods. Implementing farm-level water harvesting techniques, such as constructing small reservoirs or ponds, can ensure a more reliable water supply for barley crops during water deficit conditions. Education and extension services. Promoting awareness and providing education on drought mitigation strategies, including the use of SA and proper water management techniques, can empower farmers to make informed decisions and adopt sustainable practices. Extension services and agricultural training programs can play a vital role in disseminating knowledge and promoting the adoption of research-based solutions among farmers. Government support and policy. Governments can play a significant role in supporting farmers facing drought stress. This can include providing financial incentives for implementing sustainable practices, supporting research and development programs focused on drought tolerance in crops, and formulating policies that promote water conservation and efficient agricultural practices. By combining these solutions, farmers can enhance the resilience of barley crops to drought stress, minimize yield losses, and maintain agricultural productivity even in challenging water deficit conditions.